You know, it's something we all do, probably dozens of times a day without even thinking about it. You turn on the tap, fill up a glass of water, and just drink. We have this incredible built-in trust that it's safe. But have you ever stopped to wonder, how do we actually know? Well, today, we're going to pull back the curtain on the hidden science that protects every single glass you drink.
So yeah, let's just ask that question head on. You look at a glass of water, right? It's clear. It doesn't smell weird. It looks perfectly fine. But here's the thing. The most dangerous stuff, the things that can really make you sick, are totally invisible. So how can you really, truly be sure that what looks clean actually is clean?
And right away you can see the problem scientists are up against. I mean, trying to test for every single possible germ, every virus, every bacteria, every parasite would be, well, basically impossible. Not to mention crazy expensive. It's just not practical. So they came up with a really clever shortcut. Instead of hunting for everything, they look for one specific microscopic suspect, an indicator bacteria that tells them if something's off.
Yep, this little indicator is basically our canary in the coal mine for water. Its job is to be a warning sign. If it shows up, it's a huge red flag that other, more dangerous things might be lurking in the water, even if the indicator itself isn't the main enemy.
And here it is. Our microscopic person of interest is coliform bacteria. Now, this is a really big family of bacteria. You can find them all over the place, in the soil, on plants, and here's the key part, in the guts of warm-blooded animals. Yep, that includes us. So, if scientists find coliforms in a water sample, that is a major red flag. It's a strong sign that the water has been contaminated with, well, poop. And that means much nastier disease-causing germs could be in there too. Okay, so we've got our target, we know what we're looking for. But how in the world do you find these invisible clues swimming around in a water sample? Well, it turns out there are two classic scientific detective methods on the case, each with a totally different way of cracking it.
All right, let's dive into method 1. This is the old school approach, a kind of statistical hunt for our bacterial suspects.
It's called the Multiple Tube Fermentation Technique, or just MPN for short. And you can think of it like this meticulous three-step process of elimination. The goal isn't to see the bacteria directly, but to see the evidence they leave behind, to catch them in the act of doing what they do.
So, the MPN test has three main parts. First up is the presumptive test. This is like casting a really wide net. It basically asks, is there anything in here that eats lactose and produces gas? If the answer is yes, we move on to the confirmed test. And this step is super important, because other harmless bacteria can also make gas. So this stage gets more specific to make sure we're actually dealing with coliforms. And finally, you have the completed test. This is the final proof, the last check, to be absolutely sure about the bacteria's identity.
And the actual clue that scientists are looking for? It's incredibly simple. They take the water sample and put it in a tube with a special broth that has lactose in it. If coliform bacteria are there, they eat the lactose and, well, they produce gas. That gas gets trapped as a tiny bubble inside that little upside-down vial, which is called a Durham tube. And that's it. A bubble means it's a positive test, no bubble, no coliforms. It's really that straightforward. Now, this brings us to the key thing about this whole method. See, scientists look at how many tubes turned up positive with gas bubbles, and they use that info with a special statistical chart to figure out the most probable number, the MPN. So notice the wording there. It's not a direct count. It's a really, really good statistical estimate of how many bacteria are in the water. All right, so that's the statistical hunt. Now let's check out the second method. This one takes a totally different approach. It's less about probability and much more about a direct head count. This is the membrane filter technique, or MF. And like I said, it's way more direct. Instead of looking for clues that bacteria were there, this method physically traps every single one of them from the water sample so you can literally count them, one by one. It's just like taking a census of the bacteria. And the technology behind this is just wild. You take a specific amount of water and pour it through this special filter. And to give you a sense of scale, the pores in this filter are just 0.45 micrometers wide. That is about 100 times thinner than one of your hairs. It's so tiny that water molecules can get through, but it physically stops every single bacterium in its tracks. And the process itself is just so elegant and simple. Step 1, filter the water. Step 2, take that filter paper with all the bacteria now stuck on it and place it onto a petri dish that's full of nutrients. Step 3, stick it in an incubator for about 24 hours. And step 4, you just come back and count the dots that have grown. So, what exactly are they looking for? Well, every single coliform bacterium that was trapped on that filter grows into a visible little dot, called a colony. And on this special nutrient gel they use, these coliform colonies have a really distinct look. They have this metallic green sheen to them. It's unmistakable. So scientists just count up all those shiny green dots. And for the count to be the most accurate, they're hoping for a plate with somewhere between 30 and 300 colonies. Okay, so we have our two contenders, the statistical estimate versus the direct count. Two totally different philosophies for finding the same thing. So, how do they actually compare? Let's put them head to head.
This breakdown really tells the whole story. When it comes to speed, the membrane filter method is the clear winner. You can get results in as little as 24 hours. The MPN takes a lot longer. Then there's the result itself. MF gives you that direct, actual count, while MPN gives you the statistical estimate. But, and this is a really big but, the MPN method has a superpower. It works on pretty much any type of water, especially turbid water. You know, water that's murky and has a lot of sediment in it. That kind of stuff would just clog up the superfine membrane filter in a heartbeat.
So if you only remember one thing, let it be this. The classic MPN method gives you a probability, the most probable number. The more modern MF method gives you a direct count of what was actually there. Neither one is necessarily better. They're just different tools for different jobs.
All right, so the science behind all this is pretty fascinating, right? But let's take a step back from the lab. Why does any of this hidden science matter? Why is this work, which happens quietly in the background every single day, so incredibly important for all of us?
Well, it's because these tests are literally a cornerstone of public health. This is the science that makes sure the water coming out of your kitchen tap is safe to drink. It's how we know the beaches and lakes we swim in are clean. It's what backs up the strict regulations that stop massive outbreaks of awful waterborne diseases. This isn't just academic. It's about keeping us all safe.
And this quote really drives home just how high the stakes are. It says, one microorganism can multiply to over 1 million in approximately 4 hours under ideal conditions. Wow, think about that. A single, tiny, invisible problem can explode into a huge public health crisis in just a few hours. And that's exactly why having fast, accurate tests is so absolutely crucial. So, the next time you go to fill up a glass of water, maybe just pause for a second. Think about all that incredible science, the statistical hunts and the direct bacterial counts that are all working behind the scenes to make sure your simple act of trust is a safe one. And maybe it'll get you thinking, what other invisible systems are out there, working quietly every single day just to protect us?